Commercial rice cultivation is not just farming. It is a relentless biological arms race against highly adapted microscopic pathogens. The casualties of this conflict include the grayish-brown lesions of sheath blight and the collapsed stems characteristic of panicle blast, often called rotten neck. These visible symptoms represent the end stage of an infection. By the time they appear, dormant pathogenic spores have already breached the plant's defenses and colonized its internal tissues. To combat these microscopic threats, traditional management often relies on highly reactive blanket chemical applications. This approach drives pathogen resistance and compromises the soil ecosystem, increasing the likelihood of future crop failures. Integrated Disease Management, or IDM, transitions cultivation toward proactive ecosystem engineering. The IDM matrix consists of three interdependent layers: the nutritional bedrock, the biological arsenal, and the chemical capstone. The bottom layer engineers the host environment to establish a physiological bedrock. The middle layer deploys biological antagonists to outcompete pathogens before they infect the crop. Maximizing yield requires integrating all three layers into a single systemic defense. Host defense begins with plant physiology and the physical integrity of the cellular infrastructure. Rice blast, caused by the fungus Magnaporthy oryzae, demonstrates how nitrogen application directly influences disease vulnerability. Excessive single-application nitrogen forces rapid cellular expansion, resulting in thin, structurally weak cell walls. Adding silicon reinforces the epidermal cell walls. This creates a physical barrier that prevents fungal penetration pegs from breaching the cell. Defense also involves biochemical health, as seen in the study of Chira disease. Chira is a physiological collapse driven by zinc deficiency. Zinc acts as a critical cofactor for enzyme activation and the production of chlorophyll. Managing Chira requires the prophylactic application of zinc sulfate at 25 kilograms per hectare, integrated with regular soil testing to prevent biochemical stalling. Cultural practices like alternate wetting and drying further protect the crop by modifying the microclimate to reduce humidity. Without these nutritional and cultural foundations, baseline immunity fails, rendering later interventions ineffective. The second layer of the matrix deploys biological antagonists to actively suppress the pathogen load in the environment. Sheath blight, caused by Rhizoctonia solani, is defined by its durability. The fungus survives for up to 20 months in dry soil as hardened sclerotia, which sprout infective threads as soon as moisture levels rise. IDM uses seed treatments and soil applications of beneficial microbes to combat these soil-borne threats. One primary biological weapon is the Trichoderma species. Trichoderma colonizes the root zone and secretes cell wall degrading enzymes to directly attack and dissolve pathogenic hyphae. Another effective biological agent is the bacterium Pseudomonas fluorescens. These bacteria operate through induced systemic resistance, or ISR. Pseudomonas triggers a biochemical signal that travels through the stem, pre-arming the plant's innate immune system against future attacks. These biological controls create a hostile environment for invaders, while activating the plant's internal defense networks. The apex of the IDM matrix is the chemical capstone, which agronomists deploy with precision. Rice yellow dwarf illustrates why this precision is necessary. It is caused by Candidatus phytoplasma, which resides internally where standard topical fungicides cannot reach it. This pathogen is vector-borne, transmitted by Nephotactics leafhoppers after a latent period of 25 to 30 days inside the insect. Applying targeted insecticides like thiamoxam interrupts this transmission chain before the leafhoppers can spread the pathogen to the crop. Fungal pathogens that bypass the biological layer also require targeted fungicidal strikes. This includes specific applications like tricyclazole for blast or carbendazim for sheath blight. Timing these applications to physiological growth stages, such as booting or heading, is critical for their efficacy. Continuous use of the same active ingredients leads to the development of resistant fungal strains. Chemical tools are the final line of defense, and their effectiveness depends on being used only when other layers are already in place. Pathogenesis relies on host weakness and environmental opportunity. The IDM matrix addresses both by interlocking nutritional, biological, and chemical defenses. This layered approach compounds the effects of individual treatments. A Pseudomonas seed treatment reinforces the physical resistance provided by an optimal silicon balance. Proper plant spacing and water management further limit the humidity pathogens need to sporulate. These combined strategies significantly reduce the pathogen load before chemical intervention is ever necessary. Modern IDM prevents the widespread crop failures that have historically led to severe food insecurity. The future of agronomy focuses on engineering agricultural ecosystems where biological and physiological factors secure global yields. Resilient rice canopies are architected by utilizing every biological, physiological, and chemical tool in the matrix to out-engineer the pathogen threat.